glycolysis, and I want to preface this by saying that there's a lot of detail here that I don't need you guys to exactly memorize, such as the specific enzymatic steps. So glycolysis is really a series of enzymatic reactions that are happening one after the other. So there's going to be a substrate, there's going to be an enzyme, there's going to be a product. And that product is going to be the substrate in another reaction that yields another product with another enzyme. And that's going to happen again and again. So several enzymatic steps. I don't need you guys to memorize all the names of the enzymes or all the names of the substrates. What I need you to memorize are the main substrates into glycolysis. So for this, it's going to be one molecule of glucose and the main products that are yielded from that one molecule being broken down. So if we think about glycolysis, it's breaking down one glucose and the major yield is 2 pyruvate molecules. It's a sequence of 10 enzymatic catalyzed reactions, right? So one after the other in a sequence. This happens in the cytosol or the cytoplasm of the cell. And what we yield is 2 net ATP. And we'll talk about why it's 2 net. Because we have to input 2 ATP and we yield 4 ATP, the cost of that is 2. So we only yield a net of 2 ATP molecules. Now, there are some other substrates and products that are involved in glycolysis. One is going to be NAD+, and we talked about NAD as one of our nucleotide examples, and we said that it was an example of how we can move energy around the cell. So it's an example of storing energy that was a little bit different from ATP and ADP. So for NAD, we can move electrons around the cell. So NAD+ is going to be the oxidized form, and then NADH is going to be the reduced form. If you remember from previous knowledge, the oxidation is loss. So the oil rig mnemonic, oxidation is loss of electrons, reduction is gain of electrons, OIL, RIG. That's a good way to remember the state of moving of electrons. So NAD+, the oxidized form, and then that is reduced through the process of glycolysis to add electrons onto that NAD, and then that will become NADH+, carrying those additional electrons. And essentially, we're generating electrons that are being shuttled off to oxidative phosphorylation. So all of the electrons that we make in glycolysis and in some of the subsequent steps or stages will be sent off to the mitochondria, and they're going to play a really important part of the electron transport chain of oxidative phosphorylation that takes place in the mitochondria. Now, no oxygen is required. It's another feature that separates substrate level phosphorylation from oxidative phosphorylation. So we do not need oxygen. Glycolysis can occur in the absence of oxygen, whereas oxidative phosphorylation, as the name would imply, is reliant upon oxidation. If we look at the equation at the bottom here, we have glucose, right? We input our one molecule of glucose, we input 2 NAD+ in the oxidized state, we input 2 ADP, and then 2 phosphates, which we're going to borrow from some other molecule in the cytoplasm. On the right of our equation, at the end of glycolysis, we're going to yield 2 pyruvate as the final products, 2 NADH carrying 2 additional electrons, and then 2 ATP net per molecule of glucose. Okay, so here's a more detailed look at the steps in the enzyme catalyzed reactions of glycolysis. And so what we can see is that we put one molecule of glucose in, and we actually have to invest 2 ATP. And then we carry out the first phase of the reaction, which is referred to as the energy investment stage or energy investment phase of glycolysis. At the end of that, we're going to end up having G3P, and we're going to yield 2 G3P. So at this stage of the reaction, we're going to now have two products per substrate. So at the end of this, we're in fact going to generate two pyruvate, although we invested only a single glucose. Because in the latter half, the energy harvesting phase of glycolysis, we're going to yield 4 ATP, right? Because now we split off this G3P into two different molecules. So from fructose 1,6-bisphosphate, we cleave that into two. So now we have two separate sets of reactions occurring. And at the end of that, we end up having two pyruvate, and we end up yielding two ATP in each of these steps, which is a total of 4 ATP. But because we had to invest 2 ATP in the initial stage of glycolysis, then our net yield is only 2 ATP. And again, I don't need you to memorize these steps or stages, but I do expect you to know that one glucose is inserted. The investment is 2 ATP, and then 2 pyruvate are going to be returned at the end, or products of this, and then we're going to yield 4 ATP, but that's going to be 2 net if you subtract the cost that we invested. All right, and then here's just another look at the steps involved in this reaction. And you can see the individual enzymes that are catalyzing each of the stages of this reaction. Again, if you go to med school or grad school, you will have to memorize this step by step, which can be a real pain. But for now, I want you to know the general gist of the major substrates and major products. Let's look at the intermediary step. So this is called the linking step. And this is the transition between the glycolysis, which is happening in the cytosol, and the next stage, which is the Krebs cycle, which happens in the mitochondria. So in between that, what happens to that pyruvate? So because we generated 2 pyruvate, both of those are going to be the substrates in the linking step. We're going to invest each of those pyruvates. We're also going to invest NAD+, so we're going to have 2 more or 1 more, NAD plus in the oxidized state. That will be converted to NADH plus in the reduced state. So we're going to pick up some more electrons along the way. We're going to yield carbon dioxide as a byproduct, and then we're going to yield acetyl-CoA. So acetyl-CoA is going to be the major product that we generate at the end of the linking step. So if we look at our overall reaction, we have pyruvate that's coming from glycolysis. Remember, it's going to be 2 pyruvate. So this is going to yield 2 NADH plus because it's going to happen twice. From our 1 molecule of glucose, we had 2 pyruvates at the end of glycolysis. And so the linking step is happening twice. So pyruvate, CoA, and NAD plus oxidized. And on the right of our reaction, we're going to have acetyl-CoA as the major product. So it's going to be 2 acetyl-CoA. We're going to have carbon dioxide as our byproduct. And then we're going to have the reduced form of our NADH, and it's carrying those additional electrons. And as we said, those electrons are going to the mitochondria to take part in oxidative phosphorylation. Let's look at the Krebs cycle. So the Krebs cycle is the second major stage of glucose oxidation. So now we're working with acetyl-CoA. So acetyl-CoA was the major product of the linking step, and it's going to be the major substrate of the Krebs cycle. And the Krebs cycle has a few other names. It's sometimes called the citric acid cycle or even the TCA cycle. It's a cyclical series of enzyme catalyzed reaction. What does that mean? It means that the major substrate is also the final major product because it's going to be happening in a cyclical fashion. This is occurring in the mitochondria, and this is going to generate 1 ATP per acetyl-CoA. So if you keep in mind, we had 2 pyruvates, which mean that we're going to generate 2 acetyl-CoA, which means that for each acetyl-CoA, we're going to generate a single ATP, so that's going to be 2 ATP total. We're going to have our reduced coenzyme, so NADH carrying electrons, it's going to be 3 NADH per acetyl-CoA. And then there's going to be another enzyme. There's going to be another, nucleotide carrier, which is FAD in this case. And FAD is going to go from the reduced state, which is FAD, plus to the oxidized state, FAD plus, to the reduced form, FADH2, now carrying additional electrons. So again, FAD was another example that we looked at, which could act as a medium of carrying energy around. And so that is going to do what NADH is doing, which is picking up these electrons and taking them to the electron transport chain to be a part of oxidative phosphorylation. All right, let's look at the overall reaction and our major substrates and major products. So acetyl-CoA, remember it's going to happen twice because we have 2 acetyl-CoA, 3 NAD plus in the oxidized form, FAD also in the oxidized, it's going to be 1 FAD, and then we're going to input ADP, 1 of that, and 1 phosphate. We're going to insert 3 molecules of water. And on the right of our reaction, the major products we're going to yield are acetyl-CoA. So remember, acetyl-CoA is going to be the major substrate and major product. And then we're going to have our carriers in the reduced form. So 3 NADH carrying 3 electrons and 1 FADH carrying 2 electrons. And then we're going to have carbon dioxide as a byproduct. So we're yielding 1 ATP per acetyl-CoA, which gives us 2 ATP so far. So if we calculate how many ATP we've had up until this time, it's 2 coming from glycolysis, 2 net from glycolysis, and then 2 from the pyruvate, 2 acetyl-CoA, and so that's going to be 4 ATP so far between glycolysis, the intermediate linking step, and the Krebs cycle. Again, here's another closer look at the individual steps of the Krebs cycle, which I don't need you guys to memorize necessarily, but just have a gist of the major substrates, major products, which in this case happen to be acetyl-CoA. All right, now we can get to sort of the end culmination of all of this. So all that we have said so far, glycolysis, breaking the glucose down initially, the intermediate linking step, taking that pyruvate and making it into acetyl-CoA, and then the Krebs cycle, taking that acetyl-CoA and cyclically breaking it down to yield us essentially ATP and more of our electrons that are participating in oxidative phosphorylation. So all of those preceding steps are leading up to oxidative phosphorylation, which is going to yield us 4 ATP so far, but the majority of the ATP that we make, the majority of the ATP that keeps us alive is essentially made from this final step in the electron transport chain. So we're going to move electrons down their gradient, down a transport chain, and we're going to take the energy that's released from moving those electrons down their gradient, and we're going to capture that and harness that to make a substantial amount of ATP. Now, because this happens in the mitochondria and because of the enzymes that are used, we're going to need to invest energy to make ATP via oxidative phosphorylation. So that is another significant difference between substrate level phosphorylation, which can make ATP in the absence of oxygen. We can also call that anaerobic glycolysis for that reason, because it doesn't require oxygen, versus oxidative or aerobic respiration, which is what requires oxygen. So let's begin to describe the electron transport chain. This is really a chain of molecules on the inner mitochondrial membrane. So if we think about our mitochondria, there's an outer membrane and there's an inner membrane. And across that inner membrane is the site of the electron transport chain. Now, the molecules are going to undergo oxidation to reduction reactions. So because we have been taking all of our electrons that we have been making so far, and we've been shuttling them all off to the mitochondria to be involved in oxidative phosphorylation, we're going to be able to take that energy and move those electrons down their gradient and liberate that energy from high to low in order to use that to make ATP. The enzyme that is going to be used to catalyze the ATP production is called ATP synthase. So this is an important enzyme to remember. And the final electron acceptor is oxygen. So because we're passing those electrons along, in order to keep that chain going, oxygen must be present. This is the sole purpose that we breathe oxygen in, so that we can have it ready to accept the final electrons and keep the chain moving to make all of the ATP that we need to sustain our body and keep our cellular processes going. So the important requirements here are oxygen, the electrons that we have made so far and that we've shuttled off from glycolysis, as well as the molecule that makes the ATP, which is ATP synthase. All right. So we can think about the overall reaction of the electron transport chain and the donation of electrons. So again, think about the electrons that we've been capturing so far through the reduction of both of our carriers, NADH and FADH. And then we can also think about oxygen, which is accepting those final electrons in order to keep that chain moving. So if we insert 10 NADH carrying 10 electrons and 2 FADH carrying 2 electrons, and we insert 28 ADP in the unphosphorylated state, 28 phosphates, and then 6 oxygen, we're essentially going to yield our oxidized form. So we're going to take those electrons, we're going to remove the electrons and use them to move along the chain. And on the end products of the electron transport chain, we have our carriers back in the oxidized state now that we have removed the electrons. We have water released as a byproduct, and then we have 28 ATP, which is a significant amount of ATP. And I just want to explain the difference here. So some literature may differ on how much total ATP is yielded per molecule of glucose. And your resources actually goes back and forth on this. It goes from 32 total to 38 total. Most literature actually says 38. So I'm going to go with 38, but I just want to explain that you may see different resources that quote the total number of ATP made differently across different resources. So 38 being the total amount of ATP that we yield in a single molecule of glucose being broken down. And 34 of those 38 will come from oxidative phosphorylation, which is happening via the electron transport chain, whereas 4 of those 38 are going to be coming from the preceding steps. So 2 net from glycolysis, and then 2 each from pyruvate, or rather from acetyl-CoA being involved in the Krebs cycle, which will yield 4. So of the 34, rather, of the 38, 34 come from oxidative phosphorylation and 4 come from substrate level phosphorylation. And if you look at the other range of this, that will be 28 out of the total 32. But what I'm going to test you on and what you need to know for exam purposes is the 38 number, and then 34 of that 38 coming from oxidative phosphorylation. All right. Now, if we think about how this occurs, so just to illustrate the steps that we just described, so here's our mitochondria, which is one of our intracellular organelles. Here is the matrix. The matrix is the very central portion of the mitochondria. And then here is the outer mitochondrial membrane. And then we have an inner mitochondrial membrane, which is really highly folded nature, which we actually call the cristae of the mitochondria. Now, across this inner mitochondrial membrane, this specific surface is where oxidative phosphorylation and the electron transport chain take place. So if we kind of zoom in on that location, here's that membrane, right? The inner mitochondrial membrane. This would be the internal matrix, and this will be called the intermembrane space. So the space in between the inner and outer membranes is referred to as the intermembrane space. All right. And so what we start off with is a high concentration of hydrogen ions on the outer surface of our inner membrane, which is the intermembrane space, and then a low concentration of hydrogen ions in the matrix space. And so essentially we have a gradient, right? We have a gradient where there's more electrons out here and fewer, not electrons, excuse me, hydrogen ions. More hydrogen ions out here and fewer hydrogen ions in here. So essentially we're taking the electrons, remember we brought those electrons from the preceding steps, and we're going to take those carriers, which are carrying those electrons, and we're going to oxidize those carriers. And we're going to take the electrons off and pass them through these electron carriers, through these enzyme transporters that are inserted in the mitochondrial membrane. And so this is what we mean by electron transport chain. We're passing the electrons to this first carrier or transporter, which is complex 1. And then they're going to pass those electrons to complex 3. And then we're going to pass them to complex 4. And then we're going to pass them on to oxygen. So oxygen is standing by to be the final acceptor of the electrons. And through that process, every time we pass electrons from one to the other, we're actually moving hydrogen ions through these transporters and from the matrix space into the intermembrane space. So the whole purpose of passing the electrons along is to create this hydrogen gradient. As we pass electrons from one complex to the other, we pump these hydrogen ions from a source of low concentration to a place of high concentration, and that takes energy. Notice the color of these arrows. These arrows are red because this is moving molecules against their natural gradient, which requires energy. And the energy that we get is from passing the electrons from one complex to the other. So the sole purpose of passing these electrons is to create our hydrogen gradient. Now, our hydrogen gradient is going to fuel the synthesis of ATP because now our high concentration of hydrogen on the intermembrane space are going to move passively. Again, notice the green arrow here, meaning that this is a passive or exergonic movement, which is releasing the energy that we can take to make ATP. All right. So that is essentially how we use the electrons to make ATP through oxidative phosphorylation.